This year's prize is about how the optical microscope became a nanoscope. The Royal Swedish Academy of Sciences has decided to award the 2014 Nobel Prize in Chemistry to Dr. Erik Betzig at Howard Hughes Medical Institute, Ashburn, USA, Professor Stefan Hell at Max Planck Institute for Biophysical Chemistry, Göttingen, and the German Cancer Research Center, Heidelberg, Germany, and Professor William Mörner at Stanford University, Stanford, USA, for the development of super-resolved fluorescence microscopy. Professor Sven Lidin will now give us a short summary, please.
Thank you, Staffan. Now, this year's could someone please do something about the resolution? That's a question that microscopists have asked for many years. Ever since the invention of the microscope, this has been the challenge. We see new things, but we see that there are more things to see. In 1873, Ernst Abbe proved that the theoretical limit to resolution is about half the wavelength of the light used. For visible light, that translates to about 200 nanometers. Now, a single human hair is about 100 micrometers. That's 500 times larger than this limit. So objects of that size are very easily studied with conventional microscopy. But most of the processes in chemistry and biochemistry take place at length scales that are much smaller. A typical bacterium is about 200 nanometers across, corresponding to the Abbe limit, which means that if we study bacteria in the light microscope, they will be featureless blobs, just like our laureates right now. This prize celebrates the circumvention of the Abbe condition. And 3 scientists have contributed to break this limit in 2 different ways. Stefan Hell developed stimulated emission depletion microscopy, STED. And Erik Betzig and W.E. Mörner laid the foundations for single-fluorophore microscopy. Now, why is this important? Electron microscopy has a much higher resolution and can be used to study structures in atomic resolution. But electron microscopy does not allow us to study living cells or the processes within them. The work of the laureates has made it possible to study molecular processes in real time. It's been used to study the dynamics of transcription and translation. That is, the reading of DNA and the transference from that to proteins. It's enabled us to look at how proteins associated with disease aggregate, for example, in Alzheimer's, Huntington's, and Parkinson's disease. And it's even shown us the structural dynamic changes to neurons in the brain that takes place during learning processes. Super-resolution microscopy doesn't only tell us where, but also when and how. And that is the greatness of this development. Biology has turned to chemistry. Chemistry has turned into biology. Guesswork has turned into hard facts, and obscurity has turned into clarity. Now, could we please have that clarity back? Lovely. Now, we can observe E. coli, just like the laureates on the screen, in all the glory of super resolution, without having to kill them, slice them, fixate them, and subject them to intense radiation and high vacuum. They can be studied in real time while they live long and prosper. Back to you, Staffan.
Thank you, Sven. Now I turn to Professor Måns Ehrenberg to give a presentation.
So this prize, as you have learned already, is about seeing. And what we have in front of us is a neuron, a nerve cell, and here to the left is the way it could be seen in the old days, before the laureates did their inventive work. And to the right, we see the neuron as it can be seen today, and it's labeled with fluorescence markers, and it's this fluorescence that makes the miracle possible. And another aspect of this, which was already mentioned, is that this is a snapshot, 5-second snapshot. So you can make a film of this and see how it grows and how it moves. So what the prize is about is to extend the realm that can be addressed by light microscopy. So today, or one could traditionally look at ants with very good results. And the hair, as we have learned from our chairman of the committee, and the mammalian cell can be seen quite well. But a bacterium, you can see a little bit about the shape of it, but you can see no internal structures. Human mitochondrion, well, you can see the shape, but nothing of its internal structures. The virus, a protein, and a really small molecule, all of them will look the same in traditional light microscopy. And the laureates' work has now expanded what we can do with light microscopy from bacteria down to really small organic molecules. The first observation we can make is that the diffraction limit that Sven Lidin was talking about makes it impossible to focus a laser beam down to a very, very small spot. So here is a microscope, blue light, and here the yellow light is fluorescence. So the blue light excites it, fluorescence is coming from an organelle that we want to observe, and we have a broad yellow spot. And this has been well known, and one day this person, then a very young man, he decided there must be a way to break this abyss limit and make very, very small spots of laser beams so that we can use them to explore the finest detail of living beings. And so what he came up with in exile in Finland, actually from Germany, he discovered that if you now have your exciting blue light, you can surround it with red light, and in this way quench the yellow fluorescence from the focus. So here you see fluorescence, and here you see the quenching red light. And if you now increase the intensity of the red light surrounding the fluorescence in the middle, the fluorescence intensity gets sharper and sharper and sharper. When you really have a lot of red light, you see you can make a very, very sharp focus all of a sudden. This had been considered to be impossible ever since the microscope was invented. And then when you have this sharp focus, what you do, you let your laser beam scan your sample with an organelle, for instance, and you can scan it in X and Y and Z directions, in the side and also in the depth, and then you can make a very, very sharp picture of an organelle, which is now a mitochondrion. Huh? Okay. And here is a bacterium. It was the first experiment where Staffan Hell demonstrated his principle experimentally. 1989, William Murner, he's detected a single fluorescent molecule. That was the first time this was done. Single molecule, very, very difficult, but he did it. And then given that you could look at single molecules, Erik Betzig, he got an idea. Suppose that you have a sample here, an organelle, and now you shine light on it and you get fluorescence from it. Then the different fluorescing molecules here, they will appear like big blobs according to this Abbe's diffraction limit. But if you know that the molecules are so separate from each other that on your detector you only see light from single separated molecules, you can determine their position with very high precision. But then the problem is, if you want to label an organelle and get a high resolution structure, you need to have very, very dense labeling. Otherwise, there are big holes which you didn't label and you didn't see, yeah? Because you see through the power of fluorescence. So his idea was, let's label them with green labels, yellow labels, and red labels. And then let the microscope only see the green labels. They are separate from each other, so then you can determine their positions with high precision. The yellow ones are also separate from each other. You can determine their position. And the red ones the same. And if you put all these pictures together, you can get a reasonably well-labeled organelle. However, what you really want to have is this. And for this, you may need like thousands of colors that can be separated by the microscope. And so this didn't work practically, although it was a brilliant idea. So then one day, William Murner discovered a photoactivatable protein, a green fluorescent protein. And we see it here. It looks like a can of beer. And if you shine light 488 nanometers on it, it blinks 10 times, as you noted maybe. And then it's turned off, so nothing happens. However much light you shine on it, nothing happens. But then, if you shine another type of light with 405 nanometer wavelength, and then start to shine the 488 nanometers, it blinks again. So it has been activated. It was turned off, and it could be activated. And from this type of proteins, they started to become developed. And then Erik Betzig got a solution to his problem. And so if you now have a green fluorescent protein here, all of them are quenched, turned off, and then you send in a pulse and you activate, say, 1 per mil of them. And then all these are now separate from each other so that you can determine their positions very, very well. And then you run them until they are bleached out and disappear irreversibly. And then you activate another fraction and you determine its positions. And then another fraction, and another fraction, and another fraction, all these small fractions, and you run them out. And then in the end, you have a whole stack of pictures, and you put them together, and you get a beautiful mitochondrion with fantastic resolution. And that was the intimate work between Erik Betzig and William Murner. They didn't work together, but they could do these things in a way together. And so, here is a result from Betzig's first application of his method called PALM. So here is what's an organelle called lysosome. And here is what you see when you have diffraction limited resolution. And this is what Betzig's method gave, you see. And then this one is expanded. And here is the Abbe's diffraction limit. And you can see the single proteins here. It's a fantastic improvement. And so, from these considerations, we have awarded the Nobel Prize to these three persons: Erik Betzig, Stefan W. Hell, and W.E. Mörner.
Thank you very much, Professor Ehrenberg. We're now trying to get hold of Professor Stefan Hell, and I think we have him online from a phone in Germany. Good day, Professor Hell.
Hello, hello.
Congratulations.
Thank you so much.
So, Professor Hell, I'm sitting here in the session hall at the Royal Swedish Academy of Sciences, and we have a number of journalists from all over the world that are here. And I think some of them are eager to ask some questions. And,
sure.
Here comes the first one.
Hello, Professor. Herzliche Glückwünsche. Congratulations.
Dankeschön. Thank you.
This is Swedish television, SVT, your our live broadcast. Something has already been said about your work. Could you describe the significance of it in your own words, please?
Yes, I mean, light microscopy is very important to the life sciences because the use of focused light is the only way that allows you to see living things. However, the resolution of light microscopy was fundamentally limited to about, say, fifths of thousandths of a millimeter. And if you want to see, say, protein distributions at much higher resolution, at much smaller scales, this was simply impossible, and definitely not in live cells. Now, what we have found is that you can overcome this limit. You can see details at much, much higher spatial resolution. And this, of course, discloses how the cell works at the nanometer scale. So that's at a molecular scale, at a macromolecular scale. And this is, of course, very, very important to A, understanding how the cell works, and B, understanding what goes wrong if the cell is somehow diseased, if a disease sets in or something. So it's very important for understanding physiology at cellular level and, of course, for understanding disease at cellular level.
If I may continue,
Sure.
could you say something about how you got the idea?
Yeah, so actually the reason why I came across this diffraction problem was that I worked during my PhD thesis on light microscopy method called confocal microscopy method, but not for the life sciences, not for fluorescence. It was basically for microstructure inspection. I got bored with the topic, so I felt this is kind of 19th century physics. And I was wondering whether there is still something profound that could be made with light microscopy. And so I saw that's diffraction barrier. That's the only, say, important problem that has been left over, that people have saw that it would not be beatable, you cannot do anything about it, you have to resort to electron microscopy if you want to see the nanoscale. And so I got attracted to this problem, and I started thinking about whether there's really, say, physics or physical chemistry or chemistry to sort out the problem. And eventually I realized that there must be a way by playing with the molecules. Trying to turn the molecules on and off allows you to see adjacent things that you couldn't see before.
Okay. Thank you. Do we have some other questions to Professor Hell?
Hello, and congratulations, Professor Hell.
Thank you.
My name is Joanna Rose. I am from a Swedish popular science magazine, Forskning och Framsteg.
Right.
How big was the challenge to put the molecules on and off?
Well, it is, of course, something that you have to realize first. You have to realize that there are methods of turning molecules on and off. Now, I realized, and this was explained by Professor Måns Ehrenberg, that you can turn molecules off by a phenomenon called stimulated emission. That's a very, very basic phenomenon. Every student learns about that in the first year. And after I realized that you can turn it on and off with stimulated emission, I knew that there are also other mechanisms for turning molecules on and off that are very well described in the literature. For example, pumping things to long-lived dark states or cis-trans isomerization and so on. And so there are many, many ways of playing the same game. And this turned out to be, of course, very, very successful. So it's now a whole field and you can play it in this concept in many ways.
Can I continue? Just you mentioned actually in our magazine for about 2 years ago that
Yes, I
you
remember.
were really on the verge of giving up.
That's right. I mean, it's fair to say that the scientific community wasn't very receptive to the idea of overcoming the diffraction barrier, because people believe this barrier has been around since 1873, and the resolution is what it is, and doing something about it is, if pardon me, kind of crazy and not very realistic to do. But having realized that you do not overcome the diffraction barrier just by trying to change the waves of light, but by playing with the molecules, changing the states of the molecules, I knew that this is going to work, because I couldn't find a serious, say, physical or physical chemistry reason why it would not work out. So this is why I was so confident and kept on going, despite all the issues and problems that came up in the interim.
Okay, thank you. Do we have another question as well over there?
Sufficient Axelsson, freelance for Channel Radio and also Green Post. I think you, congratulations, and you sound still very calm, actually. So, did you expect you win the prize? How did you feel when you heard the news?
I was totally surprised. I couldn't believe it, but fortunately I remembered the voice of Professor Normark, and so I realized that this is real. But it took me a while to realize it, I must say.
Okay, thank you very much, Professor Hell, and we are looking forward to meet again in December for the Nobel Prize ceremony, and our warmest congratulations.
Thank you.
So now I turn to you. Do you have any other questions or further questions to our experts?
The question was, is there any limit that is still valid for the resolution you can get? And the answer is essentially there is no strict physical limit left. However, if you want to have a very high resolution image of an organelle by fluorescence labels, you must have labeled it properly, so you have labels everywhere, essentially. And so there may be a practical limit, but it's much lower than the limit that was set by Abbe, or understood by Abbe, 1876. It was described. Yeah? And it was so well believed, this limit. This is why Staffan Hell said, you know, I almost gave up, because he was up against the physical limit, and most people who are up against the physical limit do not survive professionally.
Okay. Any further questions? If not, I thank you all for your attention, and we hope to see you back again on Monday, the 13th of October, when we will present the Sveriges Riksbank Prize in Economic Sciences in memory of Alfred Nobel for 2014. Thank you very much, and thank you for coming.